Hello, everyone. Today I'll be sharing a brief summary of the paper, Protein Structure Prediction and Design in a Biologically Realistic Implicit Membrane by Jeff Gray, which was published in April 2020 in the Biophysical Journal. So the foundation of computational design tools is the energy function, a mathematical model of physical rules that distinguish native from non-native membrane proteins and their conformations and sequences. Currently, the most important studies of membrane proteins are the molecular dynamic simulations, which involves an all-atom lipid bilayer. An implicit solvent model enables simulations to reach longer timescales required to investigate these biologically relevant conformation and sequence changes, thus increasing the computational cost. A popular approach to overcome this increasing cost is to use solvent electrostatic models. For example, the Lazaridis implicit membrane model, which basically uses Gaussian as the solvent exclusion principle to measure the transfer energies of side chain analogs in organic solvents in order to emulate amino acid preferences in the bilayer. The authors here derived and validated the energy function parameters from experimental and computational modeling of phospholipid bilayers to capture the important membrane features. Next, they tested out the model on 4 benchmarks. Number 1, prediction of protein orientations in the membrane. Number 2, the delta G of mutation calculations. Number 3 is native structure discrimination. And the 4th parameter is the native sequence recovery. The membrane is modeled as a continuum of 3 phases, an isotropic phase representing bulk lipids, an isotropic phase representing bulk water, and an anisotropic phase representing the interfacial region. New equations were derived from these measurements, and a new energy term was calculated called the delta G membrane that computes the protein stability given by this equation. They also evaluated the ability of the model to discriminate native and non-native orientations. And also, they wanted to capture the protein stability and their changes upon mutation by comparing the different experimental values and the computational predictions. Remarkably, the distribution of amino acids and sequences designed by this particular model named Franklin 2019, or M19 in short, is comparable to the amino acid distribution in native membrane protein sequences. This results in a stark contrast to sequences designed by prior membrane energy functions that are usually over-enriched in non-polar amino acids. Future challenges involve improving the realism of the model, for instance, to include factors like asymmetric composition, non-uniformity of the membrane, lateral pressure, strain due to local curvature, and further inclusion of thermodynamic properties. Thank you.